today's exploration, we have Eli Wisdom. He's a post-bac researcher, so he'll tell you a little bit about what that means to be a post-bac researcher at Yale, and he'll be discussing how the memory of a worm works and how that might help us to learn about human memory. So it's going to be a really interesting talk. Eli, if you want to go ahead.
Okay. So are you guys seeing just the presenter mode of the screen? Great. Okay. Hey guys, good evening. My name is Eli, and yeah, I'm a post-bac researcher here at Yale, and tonight I'm really excited to tell you how we're using worms to understand memory. For the volunteers and the hosts, feel free to interrupt me if there's any questions that are popping up in the chat or you have questions. Really don't mind getting interrupted. Okay, so before I get started, I'd like to begin by telling you about my path into science. So I grew up in La Grande, Oregon, which is all the way over here on the map. And when I moved to Connecticut about a year ago, it took me 45 hours of driving through around 13 states to make it to New Haven. And this is a picture of my town, La Grande, the valley we live in. And my house is actually right here where these trees are. So I grew up really exploring the woods a lot and falling in love with nature. And another big passion of mine growing up was baseball. And I really wanted to continue playing baseball in college. And I was lucky enough to make the Pacific University baseball team. And so while I was in college playing baseball, I also started my major in biology. And it was around the second year of college, in addition to baseball, that I decided to try my hand at scientific research. And it was pretty much love at first sight. So what really struck me about doing scientific research was that I could find new knowledge out about the world, and then that new knowledge might be useful for helping develop new technologies or new medicines, and just for creating a better world. So here I am in college with my two favorite things in the whole world, baseball and research. And as graduation approached, I had to make a decision. Would I try to keep playing baseball professionally after college, or should I pursue my newfound passion of scientific research? And I thought long and hard about this choice, and it wasn't easy, but ultimately I chose to put baseball on the back burner and pursue science. So I graduated from Pacific and decided that I wanted to go to medical school and graduate school after college. But before I enrolled in any of those programs, I really wanted to get more experience working in the lab. And so I had a fair bit of research experience from college, but most of my time outside of all my classes was really spent playing baseball. So I wanted to make sure I enrolled in medical school and graduate school prepared. So I did what's typically called a post-bac. And a post-bac is basically a 1 to 2 year research training position. So here is just a chart of what's often referred to as the scientific pipeline. So typically, you would do your elementary school all the way through high school, probably go on to college and get your undergraduate degree. And from there, typically you would move on to graduate school. And from graduate school, you can kind of springboard into more scientific training and maybe stay in academia and become a faculty research director or a professor at a college. Or you can, maybe go into biotech, positions in an industry, or take government positions or leadership positions in other organizations. So I am actually in this stage right here, which is, taking a little break after undergrad to just have more experience doing research and then applying to graduate school. And I'd be happy to answer any questions any of you guys have about this, and be sure to, flow the chat with your questions about the scientific, pipeline, and it's kind of confusing, but okay. So enough about me and time for the science. So I'd like to start by asking you this question. What is a brain good for? And that's really not a great question, because the brain is great for a lot of things. Think about all the things that you do every day that wouldn't be possible without the power of the brain. The brain allows you to enjoy really good music. It allows you to solve math problems. It allows you to see my screen right now, and it allows you to hear my voice. And so really, I'm fascinated by the brain. Of all the parts in the body, I mean, the brain is really important and necessary for us to live and enjoy our lives every day. Okay, so now I'm going to ask you, does a worm have a brain? And here is the worm that we use for our studies. Its name is C. elegans, and it's about 1 millimeter long. And 1 millimeter is really, really small. It's not as small as the coronavirus or a single width of human hair. But basically, if you were to take your pencil or a pen and just make a dot in the center of a piece of paper, that's about how long this worm is. And really, these worms live everywhere. So if you guys went outside and took some dirt or maybe a clump of leaves and put some of that material underneath a microscope, I predict you'd find some of these worms. So what do you think? Does this little worm have a brain? And the answer is yes. So here is a cartoon image of what the worm's brain looks like. And you'll see it's kind of this ring structure. And the reason for this is because the brain actually wraps around the worm's pharynx inside its head right here. And so this area is kind of where the pharynx and then the intestines kind of begin in the worm. Okay, so why would we use a tiny little worm to learn about the brain? And, you know, really the reason is because compared to the human brain, the worm has a lot less of the cells that make it up. So neurons are these specialized electrical cells that are one component of brains. And the human brain, right inside of each of your heads, there's 86 billion of these neurons. But in comparison, the worm's brain only has 302 of these neurons. And so if we want to understand memory, we think it's going to be a little bit easier if we first, start by trying to figure out how all 302 neurons in the worm's brain function to form memories rather than by trying to figure out all 86 billion of the human neurons. Okay, so we are trying to use worms to understand memory. And what is memory really good for? So for me personally, I use memory to remember where I put the coffee. So when I wake up in the morning, this is how I look, tired and drowsy, and I'm looking for coffee to get me going for the day. But thankfully for my brain, which I use to store memories in, when I wake up in the morning, I remember that I put my coffee in the cupboard. And for you all listening right now, I'd like you to think about all the things that you use your memory for every day. If you didn't have memory, you wouldn't be able to remember where your favorite food is. So clearly, memory is really important. Okay, so what about the worm? Does the worm have a favorite food it remembers? And unlike myself, the worm's favorite food is not coffee, and actually the worm's favorite food is bacteria. And because the worm does not have eyes, like the cartoon worm drawn here, the worm has to rely on other senses to remember where it last ate its favorite food. And one of the ways it does that is by learning what temperature the environment was where it last had food at. And we can observe this in the lab by feeding worms bacteria at 2 different temperatures. So here are 2 worms. The blue worm had food at cool temperatures, and the red worm had bacteria at warm temperatures. Other than their prior feeding temperature, the worms are actually genetically identical twins. And so we feed these worms at 2 different temperatures, and then we put them on what's called a thermal gradient. And this thermal gradient is basically just a big slab of Jell-O that we can control the temperature on either side of. So this side of the gradient is cold, and this side of the gradient is warm. And so we put the worms on the middle of the gradient, and as I press play, I want you to observe how these worms behave. And what you'll see is that the worms migrate to the sides of the gradient that correspond to a temperature that they prefer. So the blue worm is going to go looking for food at cool temperatures, and the red worm is going to go looking for food at warm temperatures. And so the take-home point I want you guys to remember is that worms associate temperature with where to find food. And because these worms are really, really small, we can test a lot of these worms at a time. And so I'm going to play 2 videos here. And each video is just a time lapse of about 30 worms migrating on a gradient towards their preferred temperatures, the temperatures where they last had food at. So we can see worms that ate at cool temperatures migrate to the cool side, and all the worms that ate at warm temperatures are going to migrate to the warm side of the gradient. Okay, great. So we now know that worms will move towards areas of temperature that correspond to where they think they can find food. And now the question is, where is the memory of temperature stored? Do you guys have any ideas in the chat? Tell me what part of the worm's body do you think the memory is stored in? And I'll see if I can open the chat. I'm seeing some good answers come in. The brain, you guys are really smart. Oh, yeah, you guys got it. So, really, it's in the worm's brain. And specifically, we think this memory is encoded within the circuits that all these neurons connect into. And so now I'd like to introduce to you one of those neurons that scientists have found to be really important for worms to be able to remember temperature. And this neuron has a 3-letter name, AFD. And so this is what the neuron looks like. It has this cell body here, and this cell body is going to reside somewhere within the brain here. And then it also has this long neurite, outgrowth that extends all the way to the tip of the worm's head. So right here, this is the worm's mouth. Okay, and to understand how this neuron is important for the worm to be able to navigate toward its preferred temperature, we can see what happens when we remove this neuron from the worm. So I'd like to first remind you what the tracks look like from worms that ate at warm or cool temperatures. And in this picture, I just aligned all of the starting locations of the worms so that they all start at the same place. And now I'm going to show you what the tracks of worms look like that they don't have the AFD neuron. Okay, so what do you guys think is going on here? These red worms ate at warm temperatures, and we would expect them to be moving towards the warm side of the gradient. And these blue worms are just doing the same thing. Both are just kind of hanging out in the center of the gradient. What do you guys think that, this informs us about what this AFD neuron might be important for? See if I can bring up the chat. They don't remember the temperature that they ate at. Nice. That's a very good hypothesis. Nice. You guys are right on track, I think. Direction, really good. All right. Yeah, and so our conclusion has really been that this AFD neuron is necessary for proper temperature migration. So you guys nailed it. Okay, and so, based on our observation of how important AFD is for the worm to properly navigate the thermal gradient, we wanted to get a better, understanding of how this neuron is responding to temperature change. And because these worms are completely transparent, we can look at these worms underneath a microscope, and we can use some genetic tricks to express fluorescent proteins within neurons in their brain. And so here is our microscope in the lab, and we have some worms on the stage of this microscope. And here, all of these worms have a fluorescent protein that's lighting up their AFD neuron. And underneath those worms, we have a heater. It's called a thermoelectric device, but a heater will suffice. And this is just going to allow us to really change the temperature that the worm is surrounding by. And so next, what I'm going to do is I'm going to show you a video of how we change the temperature underneath these worms over time. And we have a protein inside this AFD neuron that's going to allow us to track when the neuron is responding. So I want you guys to pay really close attention to what this neuron does when I press play, and pay really close attention what it does above this red line. Okay, so we're going to start the temperature low, and we're going to ramp up over time. And what do you guys see is happening? When does this AFD neuron seem to be responding? It lights up. Nice. When does it start lighting up? Does it start lighting up when the temperature is going down or when the temperature starts rising again? Nice. Yeah. So this AFD neuron, it seems to be responding to rises in temperature. So it's really kind of this, we like to think of it as a temperature sensor. And so what we think this means, if AFD is this temperature sensor, well, let me backtrack. What do you guys think? You think is the memory of, the worm's preferred temp stored in this AFD neuron? And I'll give you a hint. Is the memory of your favorite food stored in the neurons that make up your eyes?
Eli, while they're thinking about this, we have a lot of questions in the chat about what AFD stands for. Yeah.
So AFD, all the neurons in the worm get assigned 3-letter names. I think AFD means amphid, follicular, something. I don't even know. It's really not important, so I don't think you guys should really care what AFD stands for. It's just the specific name of this neuron.
Great, thank you.
Yeah. Mm-hmm. Okay. So while you guys are thinking about if the worm's memory is stored in AFD, and let's think about what this means for what AFD is doing when the worm is moving on the thermal gradient, right? And so if AFD is this thermal change detector, it's sensing changes in temperature and responds specifically to rises in temperature. We think that when the worm moves to an area of the gradient that's slightly warmer, this AFD neuron fires. But now if the worm moves to an area of the gradient that's a bit cooler, AFD doesn't fire. And we think that AFD is this temperature change detector and that it's going to be useful for indicating the direction that the worm is traveling on the gradient. And sometimes we often refer to it as some sort of temperature compass, right? Which way does a compass always point? Right? A compass always points north. And so this AFD neuron, it always responds to rises in temperature. Okay, so now we know that AFD fires when the worm moves to warm temperatures, and it doesn't fire when the worm moves to cooler temperatures. So now we came back to trying to ask this question. What is different about the neural activity of worms that last had food at warm temps versus worms that last had food at cool temps? Does the response of this AFD neuron to temperature rises, does that response change based on the animal's preference? And here, I'm going to ask you all to be scientists in your breakout rooms. So using your knowledge of how AFD responds to temperature, work together to think about if this response changes depending on if the animal prefers warm temperatures or cooler temperatures. And so in your breakout rooms, it's a really simple activity. If you think that the neuron is going to fire, just color in the sensory neuron here, AFD green, or leave it white if you don't think it's going to fire. Hey everyone, welcome back. I hope you guys had a nice, productive breakout room session. Do any of the hosts or volunteers want to talk about maybe what their group thought up?
Sure. We had a really nice conversation in our group. Hi, I'm William. And
hey, how's it going?
Hi.
So what we talked about was that the AFD does not control the memory, because if it did, I think it's connector to the brain that tells the worm where to go. So there's an actual thing that detects this food, and then it's connected to the AFD, which then tells the brain to move or not, to the right or to the left, depending on how hot or cold the food is. And there was this other Zoom session I went to where they were using the exact same worms, and they put in a certain something that makes the worms stop moving under a certain light. So if the AFD was like it holds the memories, you wouldn't have to put in this certain kind of thing into the worm to stop it from moving.
Yeah, William, I think you're right on, man. Honestly, I kind of want your help in the lab. You nailed it. Right, when we talked about in my breakout room was kind of the same thing, that this AFD neuron, it's really only responsible for sensing changes in temperature, and it might not actually be responsible for storing the memories. Like an example we thought of was, when I was really young, I put my hand on a really hot burner, and that sucked. It was really hot. But right now I remember that burner was really hot, but the memory that the burner was hot is not in the neurons of my hand, right? That memory is in my brain, but those neurons were really important for informing me that that temperature was really hot. So I think you nailed it. I mean, this AFD neuron is kind of like that. It just is involved with sensing temperature.
Awesome. Can we have one more student share? Sorry, we had a student in our
anybody else have any ideas?
Who also had a lot of great ideas that we'd like to share. So,
Sure.
So in our breakout room, we talked about how the brain of a worm, when when it starts to explode, when it's really warm and it starts to get cold, and I asked if that happens to a human brain, and I also wondered if you cut a worm in half, then will the worm still be alive?
Great question. Wonderful questions. So the first question, right, you're wondering if, how that neuron was glowing as it was responding, is that happening in the human brain? And really what's great about worms and studying worms is that their neurons behave very similarly to the neurons of the human. And so with that neuron, when it was firing and glowing, we could just, see it glowing because we were expressing a certain protein in it, but it was responding, similarly that, your neurons do when you are remembering, one of your memories. And then the second question you had was, what happens if you cut a worm in half? Is it still alive? So there are worm species that you can cut them in half and they grow into two different worms. And that is like a totally, other cool talk that we could, spend all night talking about. But these particular worms, I'm afraid they don't do that. And they do pass away if we cut them in half. Anybody else have any comments or thoughts?
I think Layla wants to speak.
Okay, thank you. So I agree with the fact that it could regenerate if it's the brain side that's actually, if it's the brain side that isn't cut in half. But if it were the brain is way more complex. I think that it could still fire whether it's in warm temperatures or cool temperatures. So either way, despite the temperature, I think the brain would still fire whether it's hot or cold, because there are 68 million or billion neurons, you said? Plenty.
Yeah, in humans there's that many. Yeah, but you're totally right that this neuron, it actually does fire whether the worm is experiencing or the worm has a preference for warm or cool temps. So this is what it actually looks like. So the worm that ate at cool temperatures and the worm that ate at warm temperatures, they actually have the same response in the sensory neuron. Like many of you guys were saying, you guys totally nailed it. You guys are so smart. And so, you know, what does this mean about the memory of the worm's temperature if this sensory neuron response doesn't change? Do you guys have any ideas? You know, I'll monitor the chat. So what do you guys think? If the memory in this, if the memory is not in this AFD neuron, where do we, you know, where should we start looking? And if you remember, right, these neurons connect in circuits. And so, you know, there's 301 other neurons in the worm's brain. Do you think that we should start looking in those neurons for the worm's memory of temperature if it's not here in the AFD? Yeah, we should start looking in the brain. And so that is kind of where we headed next. This AFD neuron here, we know that the memory isn't stored in it, but this neuron is still important for temperature. And so we want to look at what neurons this AFD connects to. And one neuron that it connects to is this other neuron. It also has a three-letter name, this one named AIY. And now we want to see how does the response of AIY change to temperature and figure out if that response is changed if the worm has a preference for cooler or warm temps. And so I will remind you that, right, this AFD neuron always responds to rises in temperature, regardless of if the animal has a preference for that temperature. But now we were interested in how AIY responds. So remember, this is the neuron right downstream of AFD. And for an animal that prefers warm temperatures, right, they've learned that they want to go looking for food at warm temps, this interneuron also responds to, you know, rises in temperature. But what do you guys think about an animal that's trained at cool temps? What do we think this AIY is going to do? Do you think AIY is going to fire in an animal that wants to go to cooler temperatures, or do you think it's not going to fire? What do you think? It's not going to fire, I think Avez said in the chat. I think you guys are onto it. So what we found is in an animal that prefers cooler temperatures, AIY does not fire. So it's not colored in green here. And so this is really interesting that only animals that ate food at warmer temperatures have a response in this interneuron, but animals that ate at cooler temperatures don't have this response. And so we're really interested in kind of what's going on in the neurons that account for this change. Is it AFD that's not sending a signal to AIY for an animal trained at cooler temperatures, or is it AIY that's not receiving the signal from AFD? You know, what's kind of the cell biology of how this message of temperature is being sent between these two neurons, and then how that message is modified if the animal, you know, learns to prefer warm or cool temperatures. And I'll give you a hint of how we're trying to figure this out in the lab right now. And so here, I just made a new diagram showing AFD and AIY neurons. Our first guess of how AFD is sending signals to AIY is that AFD is sending them through neurotransmitters. So neurotransmitters, I just want you guys to think of them as basically little tiny chemicals that get packaged into small little vesicles inside the AFD cell, and then they get released onto the AIY cell here at a synapse. And to test if the signal from AFD to AIY is being sent through neurotransmitters, we actually blocked neurotransmitters from being released from AFD. And then we looked what happens in AIY when we block neurotransmitters from AFD. You guys have any guesses? What's going to happen when we don't have any neurotransmitters? Are we going to see a response in AIY? No. Okay, great guess. Actually, what we've seen so far is that even when we block neurotransmitters, the response in AIY is still there. So this kind of really stumped us. How come we block neurotransmitters from AFD and we still have a response in AIY? And honestly, this is the exact question I'm trying to figure out in the lab right now. And we think that the signal from AFD to AIY is coming through some other mechanism here. And so our first hypothesis right now is that, AFD is talking to AIY through what's called a gap junction. So a gap junction is basically another way, other than neurotransmitters, that neurons can talk to one another. And you guys can think of it as, it's just like a tunnel between these two cells. And so what we think is happening is AFD is sensing temperature, and then that temperature information is being sent through this gap junction into AIY. And so honestly, that's exactly what I'm doing right now in the lab today. And so I hope that, in the future, I'll have a good answer for you guys, and we can figure this question out. Yeah. So what do you guys think? Do you guys think it might be getting sent through a gap junction? No, it's not a gap junction. If it's not a gap junction, then that's really bad luck for me for all the work I'm doing right now. All right, guys. Well, unfortunately, I have to leave on a cliffhanger. We don't have the answer to that right now. But I would like to start, or I'd like to end the talk by really just reflecting on what we talked about. So began asking you all, what is a brain good for, and talking about all the wonderful things a brain can do. And, I really want to know the answer to all of these questions. But unfortunately, we can't solve, the mystery of how a brain stores memory in a single experiment. And like most of science, we have to answer all these small little questions first that build on each other. And so we're trying to, ask questions and answer questions about how the neurons in the worm allow for it to remember temperature. And although that's a small question, it's a basic, step towards answering the big question about the brain. And that's really how a lot of science is done. You ask and answer all these really small questions, and ultimately they build on each other. And then at the end, you can hopefully have a really good answer to the big questions, like what a brain is good for. And last but not least, science is done in teams. And so the questions we are trying to answer in science are far too hard for one person to work on. And it really takes the efforts of all people, of all backgrounds, and with all different, ethnicities. And so if you learned anything from tonight, I hope you take away this. Science is for everyone, and it needs everyone, and it thrives on diversity. So all of us in this Zoom call tonight, right, we see the world through different perspectives. And through those different perspectives, we can ask and answer really crucial scientific questions. And, really, my experience in science is that, we're trying to solve these really hard questions. And so we need to be able to think creatively. And it's only through the power of having a really diverse team that we can answer these questions. And so this is the wonderful lab I am a part of. And none of what I presented on tonight would have been possible without them. This was our Halloween dress up last year. If you guys have a theme that you think we're dressing up on, I'll monitor the chat. But I really want to thank you all for joining. And I hope you guys all have a great Halloween.
Thanks so much, Eli. That was a really, really great talk. During your talk, we gathered a lot of questions. They were very, very fascinated about worms. And
Great.
so I'm going to ask you maybe 2 questions. I think we have time for 2 questions before we have to end today.
Yeah, of course.
The first question, in the very, very, very beginning of your talk, we actually got a lot of questions about you and what actually pushed you to do science and what it was like balancing both, academics and athletics. A lot of people were interested in that. So I thought maybe you could talk a little bit about that.
Yeah. Well, I'll answer the last part of that, which is how can I balance athletics and academics? And really, I think I learned to do that a lot during, when I was growing up in high school. I always had a lot of classes that I needed to take care of, but I had to balance them with baseball. And I had a lot of people tell me that there's no way you can do both in college. But that really wasn't the case. And it totally is possible to play sports and, have a really, great athletic career, but also, work in the laboratory. And I was just fortunate that I had some great professors and great coaches that were able to listen to me when I told them, hey, coach, I'm working on this great experiment in the lab. Like, I might have to leave practice 10 minutes early. Is that cool? Versus my professors who would also allow me to leave class early to go catch a baseball game.
Yeah, and I think, learning to make time for the things you want to do, whether it is play sports or learn more about worms, it's up to you to do that. So for all of you guys who learn that you love worms during this talk, maybe set a couple minutes aside today to learn a little bit more about worms. Go on the internet. Maybe there's some good videos on YouTube as well about the topics, so you can learn even more. And the last question I'll ask, I will start with a wonderful joke question we've got from the group, which was, do worms know about global warming? And I thought that was great, and I had to share that. But kind of related about warming or warming of the worms. We had one student ask, is the AFD lighting up the only way for it to represent warmer temperatures, or do these neurons have other ways of communicating the information that they find out?
Yeah. So the first one about if worms can recognize global warming, I don't know if they can, but I do know this is actually interesting, and I think I'm stating this right, but basically, if there were no other living animals on Earth at this time except for worms, I think they would still take up like 25% of all living animals on Earth. So like worms are really dense in the population. So through all that collective brain power that they have, maybe they can teach us something about global warming. I hope so. And your last question about if worms have other ways of figuring out, right? Was it do worms have other ways of communicating information within their neurons?
Yeah.
Yeah. So I don't know that. I think that's a great question for science to answer. I think we think, neurons are just communicating with electrical signals, but honestly, we learn new things in the lab every day. So they totally could be using other ways of talking to one another that we just don't know yet.
Well, that's really great. That just shows that there's a lot more to learn in science. And thanks again so much, Eli, for speaking with us.